Hey guys, how's it going? I know it's been a while since I posted, but here we go. Let's hope this one goes alright. Today, I'm going to be talking about electrophoresis. I'm using the book because that's where most of the questions come from. I start with electrophoresis because you guys had it along with chromatography and mass spectrometry last week. And for those of you who are going to be in the retake, might find this useful. If you're not in the retake, then it's going to help you for the final probably. So there's going to be two videos. One of them introducing types of electrophoresis, like the main concepts and main keys of electrophoresis and how to do them. And the other video is going to be more specific, so it's going to be about electrophoresis of serum proteins, electrophoresis of cerebrospinal fluids, lipoproteins, DNA, stuff like that. So let's get started on. So first of all, what is electrophoresis? The first question on the protocol asks exactly that. What is the concept of electrophoresis? So electrophoresis is the movement of charged molecules in an electric field towards an electrode with opposite charge. What does that mean? So in order to understand this question, I'm going to have a picture displayed in the video. You're going to have a positive electrode and a negative electrode, and then you're going to have a medium, which is usually a gel. Okay, positive rod. So like you will have a positive electrode where the negative ions will go to and a negative electrode where the positive ions will go to. And what these are called, this is a bit confusing. The cathode is negative, so it's opposite. So you know cations are positive, but cathodes are negative. So the cathode is negative and the anode is positive. So you will have a medium, and then you will make an electric field through this medium by having two electrodes of opposite charges, positive and negative, the anode and cathode. So how is this? First of all, electrophoresis is a separation technique. So it's made to separate certain molecules from each other, proteins or lipoproteins or whatever. Based on what do they separate? Okay, so first of all, we will have a sample that we're going to want to separate, and this sample will have many components, and we want to separate these components. We want to see the individual components of the sample. Okay, so how we do that is the components travel in the gel or the medium at different speeds. So let's say, let's think of it that way. If you have a group of people, okay, and these people are of different heights, are of different sizes or whatever, and you want to separate them, how we could do that is we could get a net with little pores or little holes in them, and then the smaller people will go through quickly, so they will have more speed. The larger people will stop, and it's exactly the same concept, but we have another factor, which is charge. So three factors affecting the electrophoretic mobility. What does that mean? It's the speed of like the mobility of movement in a medium. Okay, so you have the three factors of mass, shape. So shape is like surface area, and charge. Okay, if you have a lot of mass, then you're most likely to be large enough to not fit through the pores, okay, or have a difficulty going through the pores, and therefore you would travel in the gel slower. And shape, if you're more spread out, then the pores, like you're going to also be slower to propagate through the medium because you don't go through the pores. And the charge, because sometimes we want to separate we want to separate substances by charge. So if you have something that is negatively charged and you have a medium that is negatively charged, so that it will repel it and it will attract the positively charged particles. So therefore, the negatively charged might travel faster than the positively charged. So what we want to do with electrophoresis is separate the molecules, like we've been saying. And how we do that is through a medium. The medium is usually one of two gels, agarose or polyacrylamide. Okay, and what we want to do with these mediums is basically make little nets with pore sizes so that we can separate the molecules depending on the size or mass or shape or charge. Okay, so on to agarose gel. So when do we use agarose gel and when do we use polyacrylamide? So first of all, a few things to know about agarose gel. Agarose gel is derived from agar. Okay, so agar is also a gel, and you will get it from seaweed, and it has it has disaccharide units. So it's made up of disaccharide units of galactose. So 1,3 linked beta D galactose and 1,4 linked anhydro alpha L galactose. But you just need to know that it's made out of disaccharide units of galactose. Okay, so yeah, one thing also to know about agarose gel is that you have anionic, so negatively charged groups in the agar gel fixed to the matrix. So that means that the positively charged ions traveling through the gel will interact with these anionic groups. Okay, so yeah, that's something to know. So that means that negatively charged will move faster or will have higher electrophoretic mobility than the positively charged ones. Yeah, so now we go on to agarose. So agarose is made out of agarobiose, which is made up of repeating disaccharide units of galactose, which has anionic groups, anionic negative groups that can attract positively charged particles, and therefore negatively charged particles will travel faster. Okay, and these repeating units of galactose, they make a structure. So the primary structure is a double helix. So like DNA, as you will see right now, and it's maintained by the presence of water. So you will have the interactions. You'll have hydrogen bonds going on between the galactose double helices and the present and the water molecules, and that will basically produce more complex structures like we're going to see here. So first you have the random coil. This is the chains of the galactose. Okay, and then you have the double helices, the double helix. Okay, here and here. So these are the chains basically interacting with each other, and this is all, of course, in presence of heat. Okay, or yeah. So when you cool it, it goes this direction, and when you heat it, it goes that direction. And then these double helices will then interact to form more aggregated structures, and then these aggregated structures will go on to form the agarose structure that we see here. And you will see the pore sizes. These are the pores that the molecules can go through. Okay, one more thing to know about agarose is that it's biologically inert, so it doesn't have any effect on the macromolecules, whether it's protein. It doesn't denature proteins, it doesn't change the structure. So what we can do after going through electrophoresis with agarose is that we can get the molecule and reuse it for more tests. Okay, yeah, so one bad thing about agarose is that it's harder, or relatively more difficult, to alter the size of the pores. So that's where polyacrylamide gel comes in. So polyacrylamide gel is more simple in terms of structure because it's made out of an acrylamide monomer, and then you have something called a cross-linking agent. So this agent comes in, you will have a polymer of acrylamide, polymers of acrylamide, like chains of acrylamide, and this cross-linking agent will form crosslinks between the chains, and depending on how the concentration of the cross-linking agent, also called bisacrylamide, you will find more crosslinks, and therefore if you have more crosslinks, you can imagine that the pore sizes will be smaller. Okay, so the ratio of monomer of acrylamide to the cross-linking agent, which is bisacrylamide, determines the pore size. So you can change it depending on the molecules you want to separate. So let's say if you have two molecules who are small in size and similar, so therefore you cannot use agarose because in that case, then they will both go through the pores and you wouldn't see much separation. The whole sample will make just one band and they wouldn't separate. But if you want to separate them with higher resolution, you can modify the pore sizes using polyacrylamide gel. Okay, you have polyacrylamide gel because it has you can change the ratio of monomer to the cross-linking agent. You can get different sizes of these pores. And so one thing that you might have seen a lot is PAGE. What PAGE stands for is polyacrylamide gel electrophoresis, and that's all that means. So do not be intimidated. One more thing about polyacrylamide gel electrophoresis is that you can have two methods of separating. You can have native or denatured proteins. Okay, so what does that mean? First, you all should know what denaturing means from the previous unit. If you don't, then that's fine. You can go back to the video, which I will do, and check what denaturing means. But what basically, in a nutshell, it means changing of the structure, or specifically the territory structure of the protein. Okay, so why would you want to change the territory structure of protein? So one thing, if you remember, if we can go back, one thing that determines how we separate molecules is shape or surface area. So if you want to eliminate that and separate them just on molecular mass, so just on the basis of molecular mass, then you would have to make them all have the same shape. Okay, so the surface area doesn't really affect the separation. And how you do that is two ways. You do it in the presence of urea. Urea, of course, is rich in NH2, and therefore it would denature by by receiving the protons, okay, or by something called sodium dodecyl sulfate, or for short SDS. So what SDS does is it's basically a negatively charged particle, and what it does, it holds the protein in chain-like structure, and we will see in the next page. But one more thing about it is that it's anionic, meaning it's negative. So it eliminates. So let's say if you have a difference in charge between two proteins, if you add SDS, it's negatively charged and it binds to the proteins in such a large ratio that you will see that the difference in charge does not affect separation anymore, because they're both very negatively charged now. Okay. Yeah, so that eliminates the factor of charge from separation, and also shape, because we said it denatures it and makes it like a chain-like structure. Therefore, the only thing that's meant to separate using SDS PAGE or SDS polyacrylamide gel electrophoresis is molecular mass. And here we can see how the SDS, if you see these little minuses, these are SDS molecules, and they basically hold the protein in this structure, and this is done in the presence of heat. Okay, that's also one more thing, because heat can aid in denaturation. Yeah, so that's the part we were talking about SDS eliminating the difference in charge. I've highlighted the parts that I think are important, so you guys can go through them and read them if you want. Slow down the video. What else? Yeah, and also, now that we've denatured the proteins, we can not analyze the whole protein, but we can analyze the subunits, so the polypeptides that make up the protein. So I'll leave this for the second part of the video, and I'll move on to capillary electrophoresis. We'll come to this later. So capillary electrophoresis, I'll have a picture in the video, so you guys should see it now. What it means is basically electrophoresis through a tube or a capillary, like it's said. So basically, it can use to separate everything that normal electrophoresis can, and it's very useful because it's very quick and not as expensive sometimes as electrophoresis. Yeah, so you can separate nucleotides, vitamins, steroids, carbohydrates, proteins, hormones, pretty much everything using capillary electrophoresis. And how it's carried out is you have the capillary, and this capillary has a diameter of 10 to 100 micrometers, depending on what you're separating, and you will have it's made out of fused silica. Okay, so silica is a glass, pretty much, okay, or sand, and yeah, you'll have this silica tube, okay, and you will have it coated with a layer of polyamide, okay. It doesn't really do anything. It just gives it more chemical strength. And then you will have at the ends of the capillary, you will have at one end a cathode, which is negative, and at the other end the anode. Okay, so there are two things that cause the ions to migrate from one electrode to the other. One of them being electrophoretic mobility, which we were talking about, but also electroosmotic flow of the buffer solution. So the buffer solution also flows from an area of less water gradient to an area of from an area of higher water gradient to an area of less water gradient, which is osmosis, as you guys already know. So the silica walls are negatively charged. So what does that say? It also has the same concept in normal electrophoresis, that you will see that the positive ions are attracted to the walls of the capillary, and therefore they will move slower relative to more negatively charged ions. So the positively charged ions will form a layer over the negatively charged walls of the capillary. So you'll see the capillary walls are coated with a layer of the positive ions. Okay, and then you will have, depending, of course, at the charge for simplicity, here we say you have negative and positive, but in true life, it depends on mass to charge ratio. So you will have a strength of charge. You have a sort of spectrum of strength of charges. So you have something positive and something less positive, and something neutral and something slightly negative, more negative, and so on. So it's a spectrum. And depending on the strength of the positive charge, you will see either the first layer of the positively charged is the highest positively charged ions, and then as you go down in charge, it starts to become less and less associated with the negatively charged capillary walls. Okay, so for capillary electrophoresis, we can also use SDS polyacrylamide gel. So we can denature and also have like sort of unify the form, so that you're only separating based on molecular mass in capillary electrophoresis. So like we said, we will have the anode and the cathode. Okay. In the capillary, you will have the sample, and this sample travels from the anode to the cathode, and right before the cathode there is a detector. So this detector detects which molecule gets there first. So like we said, this is a separation technique, so you want to see which molecule travels the quickest, and due to its properties. So if it's SDS due to its molecular mass or charge or surface area. Okay, so capillary electrophoresis can have very high resolution, especially if you're using polyacrylamide, because not only is it done in a capillary with very small diameter, but also you can modify the size of the pores in order to fit the size of the molecules that you're separating. So yeah, this is an example of capillary electrophoresis in DNA. You can have that. You can give that a read if you're interested. I do not think that it's important, but it won't harm you if you read it, of course. Okay, so the other type of electrophoresis is isoelectric focusing. not to be confused with the isoelectric focusing in gel filtration. So this is different from chromatography, okay, because this is still electrophoresis. So how does isoelectric focusing work in electrophoresis? Isoelectric focusing, of course, we know all proteins have an isoelectric point because they are made up of amino acids, and therefore they have positive parts and negative parts, okay, and at certain pH, so pH, like we said, going all the way back, is the concentration, is a measure of concentration of H+. So low pH meaning more protons in the solution, or concentration of protons, and more pH, the opposite. So yeah, at each pH, the protein will either accept or donate its H+, because they all have H+ in their structure, whether in amino acids or other functional groups. But yes, however, at this isoelectric point, the isoelectric point is a pH where the protein is neutrally charged. So it has no positive or negative charges. It's neutrally charged, okay? So what we can do is we can have an electrophoresis with a buffer, okay, of something called ampholytes. So what are ampholytes? Ampholytes are basically substances that at different charges form different pHs. So let's say when you're going from the positive to the negative, from the positive electrode to the negative electrode, okay, the charge of the gel is changing from the positive to the negative, okay, and these carrier ampholytes, they line themselves up, okay, at each charge level. And at these levels of the carrier ampholytes, you will have different pHs. So it makes a pH gradient. It makes a sort of a spectrum of pHs. Okay, you will have different pHs at each point of these carrier ampholytes. Okay, so what we can do is you can run the sample through these through the gel with the carrier ampholytes. Okay, and the molecule will stop at its isoelectric point where it has a charge of 0, okay? So at a certain point in the gel, you will have the carrier ampholyte determining a certain pH. If this pH matches the isoelectric point of the protein will stop because it will have no charge, and therefore, if it goes forward, it will have if it goes like to a higher pH, it will have more negative charge, and therefore it will go to where it can get a neutral charge, okay, so it will go up again. So it always stays at the point of the pH gradient that corresponds to its isoelectric point, okay, and that's, in a nutshell, that is isoelectric focusing. You can use it alongside SDS, so you can denature the proteins and then arrange them based on their isoelectric points, or even arrange the subunits or the polypeptides at their isoelectric points. So it's very useful, and you can use it also alongside capillary electrophoresis. So you will have capillary electrophoresis, but instead of having a uniform gel, you can introduce the pH gradient using carrier ampholytes and the buffer, and you will see that the proteins also separate depending on their isoelectric points. Okay, so yeah, that's pretty much it. This diagram shows how the gradient is formed. One more thing, you can also increase the resolution of isoelectric focusing by changing the carrier ampholytes. Okay, so the carrier ampholytes, like we said, they correspond to each pH, but we can, for example, have two pH points. If you let's say if you have two proteins with very similar isoelectric points, then you can have the spectrum go between two pHs, and the range will include the isoelectric points of both. So you will separate them because you've increased the resolution. You've sort of zoomed in on the pHs. So let's say two proteins having isoelectric points 5.1 and 5.2. With normal isoelectric focusing, electrophoresis, you will never see the difference between these proteins, and you'll think that they are one protein. However, if you increase the resolution by changing the carrier amphilites, you can form two different bands at which these two isoelectric points are, and you can sort of zoom in at narrower pH ranges. And the last topic for this video is two-dimensional gel electrophoresis. So what does it mean by two-dimensional? Okay, so now we know we can separate substances based on three things: mass, charge, and shape. Okay, so for two-dimensional, you separate them based on two characteristics separately. Okay, so the first characteristic, or the first dimension, is isoelectric focusing. So you will do the normal isoelectric focusing electrophoresis, and you will see that proteins stay at their isoelectric points, and therefore they separate. But you can also separate them based on their molecular mass. And how you do that is, well, we discussed already SDS, polyacrylamide electrophoresis, and you do that in the horizontal plane. So therefore you get separation in both the vertical and horizontal. It's almost like a graph when you're drawing a graph, for example. Just a second, get a graph paper. So in the graph paper, you will see that there is the vertical axis and the horizontal axis, and you can separate by having bands at the vertical axis or the horizontal axis. Vertical or horizontal axis, but you can also make certain points. So if I make a point at this point, it will have coordinates, okay, and it will be isoelectric. We'll know its isoelectric point and its molecular mass. So we can go to the standard. We can make a standard. So let's say we have a protein and we know its isoelectric point and its molecular mass. We can have a point of it, and then we compare that against the sample. So we see the sample at the same place. We can say, oh, this is the same as the standard. So therefore we can identify these proteins. Okay, and yeah, so the only disadvantage of this method is that you'll have to have a standard. So you will need to do this to a standardized solution first. Okay, so yes, that's the main concept of two-dimensional electrophoresis. But also, one more thing that I didn't mention: for all types of electrophoresis, you need specific dyeing. Okay, so you need to dye the proteins in order to see them, because proteins are usually not visible. If you've ever looked at a mixture of proteins in a lab, you will see that it's transparent, and therefore you need to dye it before you can do any type of analysis, especially electrophoresis, in order to see the different proteins. Okay, so you can go through the video again, and you will see that the important dyes have been highlighted. Okay, but it's pure knowledge, so I don't see a point of going through them in this video. But you can have a look at different dyes and what they're used to dye. I would recommend knowing 2 per category, and you should be okay. But for this type of electrophoresis, we're going to use either silver or Co-Massey blue. Sorry about that, guys. Okay, so we can also separate proteins with something called Western blot. So sometimes you will see that 2 proteins will have very similar isoelectric points and molecular masses. And what you can do is you can use a Western blot. So Western blot is another separating technique, and it separates proteins based on their functional groups. So it binds specific antibodies, and therefore it separates these 2 proteins. And then you can also have fluorescent antibodies, so you can see 2 different proteins because they emit different wavelengths. Okay, so yeah, and you can after 2-dimensional electrophoresis, and after a lot of types of electrophoresis, you can run them through mass spectrometry, in order to know what protein this is. Okay, so mass spectrometry will go through it next time. It's not a big topic, I don't think. And yeah, that's about it. I think that should be it for today. Well, for this video. And I'm going to post the next video with a specific electrophoresis, so like serum protein electrophoresis, like cerebrospinal protein electrophoresis, and DNA and lipoprotein electrophoresis. I also want to take this chance to apologize for not posting videos for a long time. It has been, I've had a handful of stuff happen because, as most of you know, I've been away from university and now I'm finally back. So yeah, it's nice to do videos again, and I'll see you next time. Thank you.